Afternoon. It's a pleasure today to introduce Dr. Elizabeth Kovacs to give our Barshop seminar. Dr. Kovacs received her bachelor's degree from Reed College. She then spent a short amount of time at Columbia University, but went ahead and got her Ph.D. from the University of Vermont, and then completed her postdoctoral work at the National Cancer Institute research facility at the NIH. Since then, she has been an assistant professor and has climbed the ranks to professor and vice chair of research of the Department of Surgery, director of research of the Burn and Shock Trauma Institute, and the director of the Alcohol Research Program at Loyola Medical School in Maywood, Illinois. So she's been there for almost over 26 years, and I guess she's done a good job there because she's done quite well. And I had the opportunity to visit her, and the facilities she has available to her there are extremely impressive. Dr. Kovacs works on the regulation of inflammation and the innate immune response in response to burns and tissue injury. In regards to aging, often these injuries occur in individuals who are elderly, have binge alcohol exposure, and so she's looked at the effects of alcohol and age in how a person deals with burns or severe trauma, and I believe that's what she'll be talking about today to some extent. But she's done extremely well in this area, and over the years she's published over 180 research papers. She has been past president of the Leukocyte Biology Society, and she has been extremely well funded during that time, and she now has a program project in addition to an R01, I believe. So she's done very well. I can tell you from a personal point that I believe 4 or 5 years ago you presented at an age meeting in downtown San Antonio. Is that right? I'm pretty sure she did. And she introduced. I was trying to decide if this aging thing was going to work out or not. And I went to one of her seminars, and she introduced me to the concept of. I had never heard the term inflamm-aging before. It's not hers, but she talked about it, and impaired macrophage function. And at the time, I had just taken an MD Ph.D. student, and she was becoming very interested in macrophage dysfunction in the elderly. And so we read all of Liz's papers, and we've done some stuff along those lines. And I've always thought, so I think her work is great, and I'm very excited to have her talk about it this afternoon. So with that, Liz.
Thank you, Carlos. Gosh, my mic just magically turns on. That's very scary. You guys have way more power than I think you do, these buttons and things. So thank you for inviting me here. I've actually been to San Antonio a few times, but have not been to the university. I've been to the Bandera aging meetings, and that's actually a blast for those of you who are brave enough to ride horses at a different age like this. And what I like to do is talk to you about some of my research that was initiated from a conversation with a research nurse in the ladies' room in our Burn and Shock Trauma Institute. And like many women, I use the ladies' room quite regularly. And in one of these conversations with this research nurse named Vicki McGill, I asked her what kind of patients you have in the burn ICU. And it was one of those kind of stupid conversations, like instead of saying, gee, those White Sox suck, or the Cubs are doing great, neither of those ever happen, apparently. But what I asked her was what kind of patients we have in the intensive care unit. And one year she said we have a lot of drunk patients and they don't do so well. And that started my alcohol research, which was alcohol and traumatic injury. And then a couple of years later, same research nurse, same restroom, different decade or different year. I asked again, and she said we have a lot of old people and they don't do so well. And when thinking about what kind of research projects to work on, I always like the idea of having some kind of clinical connection. I'm a Ph.D. scientist. I think it even says that up there. And my idea was not so much to just do experiments, but to do experiments that had clinical relevance. And having a research nurse tell me this in the restroom was very helpful. So by doing this, the other thing that I wanted to tell mostly to the students and postdocs here, but could also be for some of the more senior faculty, is doing something that your boss likes is a great idea. And one of my bosses at the time was Richard Gamelli, who was an internationally renowned burn surgeon. So when my lab was in, shall we say, less stable financial state, one of the things I thought of is if I did something with burn injury, he'd like it, and then he'd like me, and then he might give me some money. And some of that money that he gave me to get the first one of these two projects started was the alcohol and burn injury project, and he gave us $6,000. And even 15, 20 years ago, $6,000 was not a lot of money. But that got us going in something that has now funded our research to a great extent. We have a training grant. We've had many NRSAs funded. I've had 15 or 18 or so years of NIH research to do alcohol, and have another 10 or so faculty on our campus who are now doing alcohol, kind of because of me and kind of because alcohol is there, I guess. So the idea of doing things that are fortuitous and doing things where you actually listen to other people and then try to suck up to your bosses is always an important life lesson. Anyway, so I've always been interested in inflammation and innate immunity and the tissue response to injury. My Ph.D. research was actually at the University of Vermont, where I did lung fibrosis research. And those lungs happened to have macrophages, and I fell in love with macrophages, and then didn't do anything related to macrophages for many years, and then went back to it later in life, meaning about 15 years ago, based on just one paper by one master's student in my lab. And that convinced the NIH that I was a lung expert, which is always a good thing. But in those studies, we started with the alcohol response, with alcohol and injury, then went on to aging and injury, then worked on some studies with sex differences, and then radiation exposure. And the other life lesson to the students is in Microsoft Office, there is a technology called copy and replace, where you can take something, or editing, you can take a project that's alcohol exposure and burn injury, and you can change that to radiation exposure and burn injury and submit that to the NIH, completely different project, about 3 paragraphs of background that were completely different, a collaborator who knew something about radiation, and get something else funded. And part of my success in doing that was that there were no other people doing radiation and burn injury at the time. And there was an RFA out from the NIH saying we want people to do this because there's going to be another nuclear explosion and we need to be prepared. And my logic was, I can be prepared. I spent like 3 days visiting my mother. It was snowy in New York. I wrote the grant application as a dare, actually, from the dean's office, and it got funded, and I was one of the 2 people who was funded to do radiation and burn injury. And now we're in year 5 of that grant, and I'm not sure where we're going to go after it, but it's been kind of fun exploring a new world. So what I'm going to talk about today is aging, because that's why I'm here, I guess. And one of the reasons that I became interested in aging is because I have one parent left. I had 2 parents. My father used to say, aging sucks. And I think he was right from his perspective of aging, but not everything in aging sucks. My mother's still doing okay, but my father passed a few years ago. And what we know about aging and diseases is the reason my dad thought that it sucked is because aging increases, is associated with many diseases. There's increased susceptibility to viral and bacterial infections. There's increased mortality from those infections. Vaccine immunization doesn't work well in the elderly. There are some autoimmune diseases that are increased. Cancer is certainly prevalent. And in our world, the ability to survive an injury. And we know that some of that survival involves the immune system. And this is just a very brief overview of some of the changes of aging in the adaptive immune system as well as the innate immune system. And I'm sorry for those people who like B cells and T cells, but I'm not going to talk about that at all. The innate immune system, including neutrophils and macrophages, are where I'm going to focus our work. And some of that in the innate immune system includes the neutrophils not being as able to do their normal function of eating germs and clearing them from the environment. Excuse me. And also we're going to talk about macrophages and how macrophages are unable to perform their normal functions of producing cytokines and also kind of eating germs. And in that, what we know from previous work relating or looking at aging versus young individuals and looking at cytokine production and immunity is that relative to young adults, healthy aged individuals have increased levels of serum pro-inflammatory mediators, including interleukin-1 beta, interleukin-6, tumor necrosis factor alpha, and prostaglandin E2. They also have decreased levels of production of IL-2 and interferon gamma, and are associated with increased production of IL-4 and IL-10, and overall their decreased ability to perform cell-mediated immune responses. And when we look at this environment of having this high level of pro-inflammatory mediators leads to this phenomenon called inflamm-aging, which Carlos mentioned earlier. That term was coined by Claudio Franceschi in Italy and has become quite popular in the immunology and aging literature, because it's clear that there is an elevated inflammatory milieu in the aged individuals. And these healthy aged people could be 100-year-old marathon runners, or equivalent, they might also be 50-year-old couch potatoes. But the idea of having an elevated inflammatory response, even if you're not ill, in the elderly is pretty well represented in the literature. And if we look at this, having a balance in the inflammatory responses between what is pro-inflammatory and anti-inflammatory is important in order to have an adequate survival or ability to survive a major injury or infection. And there should be some kind of a balance in this system. But in the aged, it turns out that there's a disproportionate elevation in pro-inflammatory mediators, and in many cases not enough anti-inflammatory to compensate for that, along with stress hormones and other intrinsic factors, which result in a poor prognosis after injury in the aged. Sorry, I'm coughing into the microphone. In terms of types of injury or infection, some of the things that we and others have explored are listed here, including different types of traumatic injury. In addition, there are different types of infection models that multiple people have utilized. And also in the literature, there are a couple of models which may or may not have clinical relevance, including cecal ligation and puncture, which is essentially causing a peritonitis-like disorder by tying off a portion of the intestine and poking holes in it with needles. And another model which many people use, we've used but don't really like anymore, is lipopolysaccharide or endotoxin administration. And part of what we're thinking about in terms of trying to do some of these animal models and trying to mimic what happens in people is to try to come up with something that's clinically relevant. And the clinically relevant scenario that we came up with is burn or scald injury. And in going to meetings at burn meetings as well as others, one of the things that we've seen is that this is something that could happen to young people as well as old people. And there are very often scenarios where an elderly person is cooking in the kitchen and a horrible phenomenon called a granny burn, where the individual touches the flame of their stove and burns one side of their body because they're wearing a loose house dress and is unable to get away from that fire. So scald and burn injuries are among the things that we work on, for better or worse, and that was the clinically relevant scenario that we came up with. Although we could look at other things, and in collaboration with Carlos, we did some things with infection, respiratory infection, as well as combinations of burn injury and respiratory infection, which I'm not going to have time to talk about today. So the etiology of burn injury is shown here, that about 450,000 people per year in the United States sustain burn injuries that are severe enough to require medical attention. 45,000 of those result in hospitalization and about 3,500 deaths per year. And if we look at the mortality of the elderly in this scenario, that 65%, people this should be over 65 years of age, account for only 12% of the burns in the United States, but seem to have a disproportional increase in mortality. And back in the 1970s, comparing the mortality of individuals who are under 65 years of age or over 65 years of age resulted in the 70s of about 20% of the younger people passing on and about 50% of the older people passing on. And as of 2012, we've been able to reduce these to some extent. Somebody who is young-ish, under 65, I'm still under 65, so I guess I'm young, such as it is, it would only have a 1% chance of dying from that injury, whereas somebody over 65 has an 18% chance. So it's still disproportionately high in the aged population. So why is that? And some of the observations are pretty obvious to most of us. That one reason is that the elderly are more likely to have pre-existing conditions. And some of these disorders, diabetes, cardiovascular disease, et cetera, alter how the immune system works and alter your ability to survive a major injury. In addition, this phenomenon of inflamm-aging occurs. And that suggests that, or data show, that aging alone increases the risk of morbidity or mortality after burn injury. And I'll show you some clinical data at the very end if you guys don't get sick of me. So what are the causes of death after burn injury? And among the causes of death, multiple organ failure, where literally your multiple organs fail, starting with the lung, which we'll talk about in a minute. In addition, sepsis or an infection throughout the body, and acute respiratory distress syndrome, which is a pulmonary response to traumatic injury and other insults. And if we look at the burn population, the patient population, most of the burn patients don't die from their primary injuries, like burn injury to the skin. They die from systemic complications that result from that burn injury. And those systemic complications include inflammation throughout the body. And that if you think about it, the way this works, or the way this can work, is that there's excessive release of pro-inflammatory mediators into the circulation. And we'll talk a little bit more later about how that can occur. And that from that, there's increased mediators that circulate to other organs through the circulation. And that can cause damage to otherwise uninjured organs, which results in organ failure. And as I said, the lung is often the first organ to fail. And the reason that it's it is associated with failure, we'll talk about in just a second. But it is clearly the first organ to fail, that about 45% of the patients show some kind of damage to the lungs, burn patients, even if they don't have smoke inhalation injury. So there's something about the peripheral injury that has a pulmonary complication. This was my license plate when I was in graduate school at the University of Vermont. It cost $5 a year to have vanity plates there, so it was a pretty cheap thing to do. And since my car was always in the parking lot, everybody thought my boss, who was a pulmonologist, was the person there all the time, but it was me. He didn't have vanity plates. And unfortunately, this says 1984, so you can see when I was in graduate school long ago. So why is the lung as an organ that is injured from remote injury? And the lung is a very cool organ. There are about 40 different cell types in the lung, which means you can do lung research forever in your career and keep doing it because there are other cells to look at. But the reason that it is an organ that gets injured is for several obvious or not so obvious regions. This is a histologic picture of the lung. The air is in this empty area, and these are the walls of alveoli, which are just chock full of capillaries. This is the site of gas exchange. And oxygen has to go from the lumen here in the alveolus through the wall into a red blood cell that's cruising through one of these capillaries. And it's important to think about that a reason that the lung is injured, in part, is that all of the blood that returns to the body from different parts of the body is pumped from the heart to the lung, and the lung is where it gets reoxygenated. So there is a direct route back from multiple organ systems, and this is the first capillary bed that activated white blood cells will see, all of these capillaries that run through here. So there's a very large surface area, increasing the probability of damage through these vascular channels. There's a very delicate wall architecture which allows gas exchange to occur, and if this gets thickened with edema, then obviously gas exchange isn't going to occur very well. There are many macrophages as the primary resident inflammatory cell. Some of these hang out in the lumen or in the alveolus, and some are in the interstitium or in the connective tissue adjacent to those capillaries. There's also potential for invasion of infectious organisms, both from the airway coming in as you breathe in, as well as from the vasculature running through all of these capillaries. So it's a very delicate organ which obviously needs to be maintained in this structure for oxygen transport to occur efficiently. So we first asked the question, are aged mice less able to withstand a burn injury relative to young mice? And in order to do this, we also had some pulmonary-related questions. If this occurred, does it correlate with more pulmonary inflammation, and is there aberrant production of pro-inflammatory mediators in the lungs? And to do this, we used a mouse model using C57 black 6 female mice. We've also done this in male mice and see pretty much the same thing. And our young mice were 4 to 6 months of age, and our aged mice were 18 to 22 months of age. A Balb C mouse, at least in our world, the oldest Balb C mice that we ever have worked with is about 24 months, and that correlates with a rather old human. For these studies, we had to anesthetize and shave the mouse, and then exposed 15% of its total body surface area to either scalding water, boiling water, or sham injury. That 15% total body surface area is about the size of a quarter on the back of a mouse. So it's not a very big surface area. But if you think that mice have big ears and tails, that turns out to be a reasonable size injury. And then at various time points after injury, we examined lungs for a variety of inflammatory parameters. We collected blood because it's available, I guess. And we we monitored survival. And for those of you who I've talked to already, one of the things that we're very good at is called freezer science. And that is when we say we're collecting the lungs and blood, we actually collect several other organs, and we put them in the freezer for a rainy day. And our first year of phenomenal productivity was a year that we had to kill every mouse we owned because there was a mouse hepatitis virus outbreak in all of the Chicago medical centers. So we did a lot of freezer science. We had all of these samples. We ground lots of things up and measured lots of things and published 12 papers that year, not doing another single mouse. So it was very productive in one regard. We emptied the freezers before they thawed for some other reason, and we were able to do this. So when I say that we're only collecting blood and lungs, I lie. But in any case, in monitoring some of these treatment groups, we ended up having 4 experimental groups: young sham-injured mice exposed to room temperature water. Young burn exposed to boiling water, just that 15% of their back. Aged sham and aged burn. And from these studies, we looked at mortality initially back in year 2000. That was the mouse hepatitis virus year. So we had to kill everything or let them kill themselves, I suppose. And in these survival studies, what we were able to do was look at the percent survival over time. And we found that young mice, given this small scald injury, all did fine. And the aged mice succumbed to their injuries. So about 70% of the mice died from just this injury alone after about a week. And if we looked at the lungs of these mice at early time points, like 6 hours after injury, before pointers, about 6 hours, at 6 hours after the injury, we see that the sham animals, the young sham animals, have this delicate alveolar architecture, which I showed in the initial picture, and aged sham animals have that lovely delicate architecture. But at 6 hours after injury, the lungs of young burned animals don't look so hot. There's thickened alveolar walls, more leukocytes in those walls. And in aged burn, they also don't look very, very good. And in fact, the aged look worse than the young. And if we wait till 24 hours, a more convenient time point for most of us, we see that the young burn animals recover most of their architecture to normal or very close to normal, where the aged still don't look very good. There's an accumulation of leukocytes. You can see that hopefully this doesn't look as clean or as good as it could, but is an improvement over the earlier 6-hour time point. And if we look at the cells, or look at the cells that could be in that place, in the lungs we could see macrophages like this alveolar macrophage just hanging out at the edge of an alveolus. And that's the predominant cell type in the alveolar space. And in addition, we may see some infiltrating neutrophils with this type of morphology as a response to acute inflammation. And those are the cells that we kind of focused on. And in this high-power micrograph, we can compare at this 24-hour time point the young burn to the aged burn, and we can see that the architecture of the lung is much better or much more normal in the young burn than it is in the aged. And you can see there's thickness of the alveolar wall, which hopefully you can see pretty clearly, as well as at the edges of the arrows. There are leukocytes, in this case primarily neutrophils, that are present with the alveolar wall. We think that they're in the alveolar, either in the alveolar wall or in the blood vessels in the wall, and we are still working on trying to figure out exactly where they are, and I'll explain that a bit in a minute. So to further identify these cells, we used immunofluorescence to try to identify neutrophils, which we refer to as GR1 positive cells. We've also identified macrophages, which, well, neutrophils are green, will appear green, macrophages red with MoMA2 staining, and showing immature monocytes, which are GR1 positive and MoMA2 positive, where the two colors merge. And we used Hoechst stain as a nuclear marker. And I don't know, hopefully you can see this reasonably well, that the alveoli of at 6 hours, the alveoli of the young sham animals and the aged sham animals have just a few leukocytes present. Here are a couple of macrophages in red and maybe a neutrophil or so. And at 6 hours after burn injury, we see lots of neutrophils accumulating in the young as well as in the aged lung. But if we look at 24 hours, we see that some of that has repaired or cleared itself out. In the young burn, we see very few neutrophils present, but in contrast, in the aged burn, we see lots of neutrophils present. And if we quantitate those cells, looking at the number of the cell population that is the GR1 positive, MoMA2 negative, which are the neutrophils, by counting these cells, we're able to see that after 6 hours after burn injury, there's an increase in the number of neutrophils in the lung in young as well as in aged, and that's significantly elevated over the shams. And at 24 hours, the young has returned back to normal levels, whereas the aged is still elevated. So having these elevated number of neutrophils in the airway could be potentially good or more likely potentially bad. We wanted to find out where those neutrophils are and what they're doing. And the three places where they probably could be, if they're sort of in this neighborhood, are they could be in the airspace in the alveoli, they could be in the pulmonary capillaries, or the blood vessels that run through that wall, or they could be outside the capillaries, but still within the connective tissue that lines the airway. And to try to figure out, and their location kind of depends on where the insult is. Usually if you give an insult through the trachea or through the airway, you would expect more of those neutrophils to transverse or migrate into the lumen, where if you have an insult that comes from the blood side, you would probably expect them not necessarily to leave the blood vessels, but maybe to be within them. And this really only matters in terms of therapeutic intervention. If you want to give something that will decrease the number of neutrophils that are there, if they're all in the airway, you might want to give something by aerosol. And if you're trying to do something that might help the whole body, you might want to give something systemically through the vasculature. So to do this, we perform flow cytometry on bronchoalveolar lavage cells and stain those cells for the macrophage and neutrophil markers. And looking at these cells as the percent of cells in the lung lavage, we see that the macrophages are the predominant cell in the airway, in the airspace, with somewhere between 70-something, 72, and about 85% of the cells in the young and aged animals, regardless of injury or macrophages, with just a few neutrophils in the airspace. So not very many of those neutrophils are getting over to the airspace. Most of them are somewhere else. But in spite of that, there's an elevated level of one of the neutrophil chemoattractants, Kc, in the lung tissue. Here measuring the amount of Kc protein in young and aged subjects at 6 and 24 hours. That at 6 hours, we see a mild elevation in Kc in the lungs of the young burn-injured animals, and we see a much more marked elevation in the lungs of the aged burn animals. At 24 hours, we see a return to almost normal levels in the young burn, but we still see an elevation in the aged burn, suggesting that there's still some kind of inflammatory event going on in those lungs. So a summary of this part so far, compared to young burn-injured mice, aged burn-injured mice have decreased survival. I didn't get a chance to show you our oxygen saturation studies, but we also see decreased oxygen saturation in the periphery. We see elevated pro-inflammatory mediators in the serum and in the lungs, and we see neutrophils accumulating that we could measure either in lung tissue by immunofluorescence or isolating them in lung homogenates, which I didn't get a chance to show you. And that from our lavage studies, it's pretty clear to us that these neutrophils are present in the lung tissue, but are not have not made it all the way to the airway. Yes?
I was curious about the macrophage numbers in the lung. Did you quantify them as well?
Yeah, I showed that a couple of slides ago. These are macrophages.
The airspaces, but what about?
This is from bronchoalveolar lavage. So this is squirting saline into the trachea and sucking out what's there. So this is what's in the airspace. And if we look at the number of macrophages in that immunofluorescence, we didn't count them because they were pretty much the same. Oh, I didn't show that because they're the same. Sorry about that. Yes.
So, what in the mouse, I guess the equivalent of the burn degree or depth is, like in a human's first, second, third degree burns. So we're talking about full thickness burns.
This is a full thickness burn. This is a full thickness burn. The tissue is necrotic. Yes.
Okay. The other question I have is about the infiltration of mesenchymal cells into this at 24 hours, let's say. Did you see, or was anybody looking to see whether or not that had anything to do with the inflammatory processes going on?
One of my current graduate students is looking at mesenchymal stem cell numbers, not so much infiltrating new ones, but the resident ones in the lung. But I don't have any convincing data. And actually, she's not working on aging. She's working on our alcohol project. But she does see changes after burn injury, and she sees changes in response to alcohol exposure. Okay, thank you for those questions. So one of the things that we wanted to do was figure out ways to decrease this inflammation. And we wanted to do it in a very specific manner that would only decrease the inflammation in the airway, but not alter things like cutaneous wound healing, because we have a relatively large quarter-sized burn on the back of the mouse. So other strategies that we've used in our lab to block either cytokines, chemokines, or inflammatory responses are shown here. One could block or give an anti-cytokine or anti-cytokine receptor antibody to try to block the inflammation. One could give an anti-chemokine or chemokine receptor, give anti-inflammatory cytokines themselves, perform studies in genetically deficient mice, and as I'll mention a little bit later, we did a lot of things with IL-6 deficient animals. Those were actually among the animals we had to kill when we had our mouse hepatitis virus outbreak. So we were aging knockout mice, which made the experiments very expensive and very depressing, since we weren't able to age them to the old enough age to have these studies performed properly. We've also given low doses of glucocorticoids to block inflammation, as well as low doses of 17-beta-estradiol. And just about all of these studies were sort of, in those days, targeting IL-6. But in the present study, we decided, or I guess I was going to talk about the IL-6 knockout, but I kind of already did. The two that worked the best were things that would block IL-6 either directly or indirectly. Directly by not having any, and indirectly by using low-dose estrogen, which turned off IL-6 levels in our animal models. And for these studies, we did many of them after burn injury, some after LPS or endotoxin administration, and found that doing either of these treatments would decrease inflammation, would increase cellular immunity, and most markedly increase survival. But in the present study, what we're trying to do is decrease lung inflammation specifically, and we decided to target or use an anti-chemokine antibody. And the chemokine chemokine receptor antibody, and the chemokine we were going after were the neutrophil chemoattractants. And by giving an antibody against CXCR2, which is the receptor for MIP2 and KC in the mouse, we wanted to see if that would decrease immunity. And to do this, decrease lung inflammation, we decided to pretend that we were EMT response people who would rush to the scene of a fire or something and see that an elderly mouse was in the fire and say that we've got, you're old, we could give you this antibody and it might be able to save you. So instead of trying to do anything before the injury, we elected to do things at 30 minutes after the injury. So something was already beginning to ferment in these animals, and we're trying to target that time point. And if we look at the lungs of anti-CXCR2 antibody-treated mice, given the antibody intraperitoneally, comparing IgG to anti-CXCR2 antibody, we see that the lungs of the IgG-treated aged burn mice had that impressive lung inflammation going on. And we treated them with this antibody 30 minutes after the injury. It decreased inflammation in those airways. And if we look at the lung neutrophil concentration in these animals, we see that in the aged mice that are given IgG, there are a lot of neutrophils, GR1 positive, MOMA2 negative cells in the airway. And if we give that antibody, we knock that down to a much more reasonable amount that makes the lungs look better. And we look at, just to summarize some of those data, some which I couldn't show you because of time, so we can get on to other things. Part of what we're trying to do is compare these aged mice given burn injury with anti-CXCR2, and compared to having no antibody, treating them with the antibody decreased lung inflammation. We saw no changes in either KC or MIP2 levels, but important to us that we saw no change in the neutrophil recruitment to the skin, where we saw another site of injury that needed to repair. So there was no change in neutrophil recruitment there. Survival studies are still ongoing, and we haven't yet performed studies, or are now performing studies looking at oxygen saturation. Because in my opinion, it's great to fix something histologically, but if it doesn't improve the function, I guess who cares? Or I don't care as much, I guess would be a better way to put it. So important to us, why didn't neutrophils from those aged mice migrate? Why did they stay in the tissue? There were chemokines present. There was lots of Kc as one of the neutrophil chemoattractants. So we wanted to know if neutrophils are able to respond to chemokine signals if those cells come from old mice, and do they express chemokine receptors? Are they functional? Are they able to migrate? And to do this, we used a modified Boyden chamber and measured neutrophil chemotaxis in vitro initially. And we had chambers with media. We had a polycarbonate filter, media versus Kc, and we used cell tracker green-stained neutrophils so we could watch them migrate in response to those chemoattracting signals. And when we looked at this initially, we saw that looking at the percent of cells that migrated, we saw that there's a difference in the migration ability, even in the absence of stimulation, in young and old. That cells from old animals seem to migrate. They seem to just move around. And we found that when we gave Kc over a very wide range of chemokine, up to 10,000 picograms per mil, we saw no difference in the ability of those cells to chemotax. We got to this peak pretty quickly and no additional chemotaxis. In the aged, we found that there really wasn't any chemotaxis to begin with. They just sort of flopped around and didn't migrate very far. And if we compare that to a standard control of FMLP, we saw the same pattern of the aged cells are more migratory. And if we looked at this, normalizing it to the percent of unstimulated cells, we see a pretty marked difference that cells from young chemotax at a very low level of chemoattractant, and then that's as far as they can go. But the cells from the age just don't chemotax at all. They don't really migrate very far. And we compare that to our colleague Janet Lord at the University of Birmingham. She saw something similar in chemotactic index of neutrophils obtained from the blood from young subjects under the age of 35 versus aged subjects of over 60. That those cells from the aged subjects didn't chemotax very far. They didn't migrate very much. But in contrast to their ability to migrate in a directed manner toward a signal, they were able to move around in the dish, so their chemokinesis was good or equal to that of neutrophils from young subjects. So neutrophils can move, they just don't know where to go if they're from aged subjects. And we also did an in vivo study in which we injected the chemokine Kc subcutaneously in mice to try to monitor the ability of neutrophils to chemotax toward that signal. And in doing that, we were able to see that neutrophils from young mice, isolated from the blood, were able to chemotax very well. But even at higher concentrations of chemotactic signal, neutrophils from aged mice only chemotaxed about half as well. So there's something in the neutrophil that decreases its ability to migrate. So some of the reasons why neutrophils can't migrate include things that are shown here, that there may be a dysfunction in the ability of intracellular signals to activate that pathway. We've seen, or others have shown, that decreased mitogen-activated kinase phosphorylation occurs. There may be changes in other signaling pathways, and we know that there's definitely a decrease in neutrophils' ability to phagocytose, which sort of goes along with the same kind of process, since many of the signaling pathways of cell migration, actin polymerization, and migration are similar in migration and phagocytosis. Receptor desensitization is also another possibility, as well as changes in receptor expression. And I'll just show you a little bit of our data on receptor expression. Looking by flow cytometry in the blood of aged mice at 24 hours after burn injury, looking at the percentage of CXCR2 positive neutrophils, turns out that it decreases in the aged mice. Looking at the young mice versus the aged mice, looking at CXCR2 over at GR1, we see that there's really no difference between the two populations in the absence of injury. And when we look in the presence of injury, we see that the burn-injured animals have lower expression of CXCR2. And when we look at that in a quantitative manner, although not utterly dramatic, there is a decrease in the number of CXC, or the mean fluorescent index of CXCR2 on the GR1 positive cells. So there's something different in the processing or the activation potential if there's less receptor present. So another thing that we started looking at were adhesion molecules, and I just have one piece of data on that, that the two things that you need for neutrophils to migrate are having or chemokines present and having adhesion molecules upregulated. And one of the quick questions we're asking is maybe there's some difference in the pulmonary vascular endothelium that might make those cells adhere more. And looking at the percentage of cells that are co-express ICAM, the adhesion molecule, and PCAM, an endothelial cell marker, we see that there's a marked elevation of expression of ICAM in the vasculature of aged burn-injured mice at that time point. So to summarize this part, compared to neutrophils from young mice, we see that there's an increased capacity to migrate in the absence of a stimulus, at least in vitro, and that there's no further chemotaxis in response to a chemoattractant like KC. We also found that there's a decrease in CXCR2 expression in blood neutrophils after burn injury, and that CXCR2 expression in the lungs in the absence of injury is present in the absence of injury with no further decrease after burn, which I did not get a chance to show you all of. So we thought of some other things to begin to examine. As I mentioned, we looked at endothelial ICAM expression and started to look at some of the cytokines that are present in the lungs as well as in the blood of burn-injured mice. We see that the lungs have an increase in IL-1 beta, which I didn't get a chance to show you, which could be a cause or a mechanism by which ICAM is upregulated. In addition, our most dramatic changes are seen in IL-6 levels, which are elevated in both the lungs and the blood of aged mice, even in the absence of injury, and are then further elevated after burn. So this IL-6 information made us start thinking of where pro-inflammatory mediators come from. And one of our thoughts is that if we have high levels of pro-inflammatory mediators, they must be coming from macrophages. Because we like macrophages, we have to think that way. And we wondered whether inflamm-aging was really macrophaging. So we started looking at the spleen, because the spleen is, as I've told the few people I've met with today, the spleen is an easy place to get lots of macrophages. So one of the postdocs in my lab started collecting the spleens from our aged mice with and without burn injury, collecting those cells and culturing the splenic macrophages with LPS as sort of the standard generic stimulus to see whether cytokines were changed. Uh-oh, am I going the wrong way? Yes, I'm going the wrong way. So what we're able to see is that after burn injury, sham-injured young animals, where their macrophages are given LPS, have an elevation in IL-6, as shown here. And young animals that are burn injured produce a higher level of IL-6. So something in the burn environment of the young sort of pre-activates those macrophages so they can produce more cytokines in response to LPS. In the aged animals, we don't see that. In fact, we see less of a level of elevation, although in this case it's not significant, in the sham-injured animals, comparing this bar to over here. And after burn injury, we don't see that very robust activation of IL-6 production. So that made us think there's a discrepancy between what we see in vivo, assuming macrophages are the cells making the cytokines, and what we see in vitro. So when compared to young animals, whether it's humans or mice, aged animals have higher levels of IL-6. That's been reported by a gazillion different labs, and that occurs in vivo. But production of IL-6 and other cytokines by macrophages in response to LPS stimulation seem to be lower. And from these studies, we then said, well, we better look in the absence of injury. So another member of my lab took macrophages from the spleen of mice that were uninjured, and my pointer keeps having problems, and looked at the level of IL-6 production and saw that cells from young animals were able to produce a high level of IL-6, and cells from older animals produced less IL-6. So there's some difference in vitro, even if those animals are not injured or anesthetized or anything else. And this isn't just IL-6. We saw the same phenomenon with TNF. Yes?
Do you know if it's that fewer cells are producing, or that the ones that are producing less?
That's a good question. I have no idea. All we know is that what we have is the readout, at least so far, from these studies. When we looked at some of the signaling pathways, overall, pointer. So using LPS as our stimulant, we saw this sort of global concept of all of the pro-inflammatory cytokines were lower. And we found that this was not dependent on the concentration of the ligand, the timing of those studies, or the source of macrophages, which in those days to us were splenic, peritoneal, and blood monocytes. And that all of these cells produced less IL-6 and TNF and other pro-inflammatory mediators in response to toll-like receptor ligands. But burn injury is not the same as LPS. So, of course, we had to see what would happen if we gave an aged mouse LPS. Back in those days, we did those kind of things. And we found that just like our previous studies with burn injury, that young animals given LPS were able to elevate their circulating IL-6. Aged mice had high IL-6 to begin with, and I've talked to a few people about possible sources of that, and we can go into that later if we have time. But if you look at the level of IL-6 that's produced in aged mice given LPS, it's about 5,000 picograms per mil, way above the about 100 that is generated in the young. So there's something about that in vivo activation that makes very high levels of cytokines. Unfortunately, for our sake, it didn't look like inflamm-aging was the same as macrophaging, because what we were able to see is that from our in vitro studies that we saw lower levels of these cytokines being produced. We sort of chased that to a signal transduction pathway and found decreased phosphorylation of a couple of the MAP kinases in those studies. And interestingly, which we'll get back to later, again, if we have time, that we saw elevated IL-10 after LPS stimulation in this cell population. So that told us that there's a defect in macrophage activation if you give a classical kind of macrophage activator. And many people have been studying the macrophage polarization phenomenon, which I'll refer to as a phenomenon, because it's really just something that's driven by extracellular signals. That there's classical activation to what's referred to as an M1 macrophage, and alternative activation to an M2 macrophage. And the classical activation includes things like lipopolysaccharide as a stimulant, or a combination of tumor necrosis factor alpha and interferon gamma using whopping doses of TNF, which is one of my concerns about it. Alternative activation involves using IL-4, IL-13 as the stimulant. And the readout for some of these systems include a series of pro-inflammatory mediators or an anti-inflammatory mediator and some other weird genes that people seem to have associated with M2 phenotype. And what's overall important is that the M1 classical activation leads to the ability to kill bacteria, and the M2 phenotype, which is now subdivided into A, B, and C, and who knows what else by now, each week there's a new category, I guess, are sort of in general the cells that are anti-inflammatory or the cells that are pro-fibrogenic or pro-wound healing. And in looking at this, we wanted to know if there was an alteration in the M1, M2 phenotype in macrophages from the spleen of mice. And we were sort of predicting that if you've got a pro-inflammatory environment with these low basal levels of pro-inflammatory cytokines in the age, we should see more of an M1 phenotype. That kind of makes sense. Alternatively, I guess if you're a positive person, that's the way you'd think. Alternatively, you could say that the continuous exposure to these pro-inflammatory mediators would downregulate a response. That's the negative. So lower levels of an M1 cytokine. And we know from our work that we see a decrease if we have the TLR signaling, or toll-like receptor 4 signaling, and 2, I think are the two we worked on, we see a decrease in at least the pro-inflammatory cytokines, which would be a decreased M1 phenotype. And what we saw from these cells, looking at IL-6 gene expression by real-time PCR, is that if you give IL-4 as a prototypic M2 activator, we see very little IL-6 being produced. In contrast, when we give lipopolysaccharide, the lipopolysaccharide induces a huge level of expression, about 200-fold induction of expression if the cells are from young mice, but only about 50-fold induction if they're from the aged mice. So just like the previous studies looking at cytokine levels, we see that the LPS does not induce a lot of cytokine message. And if we give this sort of sledgehammer of TNF and a normal-ish level of interferon gamma, we see production of IL-6, but not as much in the aged cells, which is a significant reduction. In contrast, if we give those cells IL-4 and look at an M2 marker, one of the M2 markers, which is up here, arginase 1, we see that there's a much higher level of production of arginase 1 in the cells from young animals relative to cells from aged animals. And we also see lower levels of activation with other stimulants. Another M2 marker, Fis1, shows that same kind of pattern with IL-4 inducing a lot more than in age than young. So this sort of tells us that looking at splenic macrophages, that there's a shift, that what we see actually is not a shift from M1 to M2, but rather that there's a suppression or decreased expression of both M1 and M2 markers in splenic macrophages from aged mice compared to young. Kind of a global immunosuppression, so those cells can't really do anything. They're almost like anergic. One of the things we wanted to see if this was compartment specific, again, looking at other sources of macrophages, we grew out bone marrow macrophages. This, I shouldn't say wasted, but consumed a lot of one of my postdoc's time trying to grow out macrophages, getting macrophages, and hitting her head against the wall trying to show differences in age. And she found that there were no longer differences in age because those cells no longer were able to show age-dependent changes. So, of course, we want to get back to the lung since that's my favorite inflatable organ. And what do we see in aged macrophages, or macrophages from the lungs of aged mice? So we did 2 different kinds of studies in the lungs of our aged mice. Most of our studies so far have been in unmanipulated animals, but we've also looked a little bit at burn and sham-injured animals, and I'll show you just a bit of those data. So again, we took lungs, we lavaged them, we collected the bronchoalveolar lavage cells, which again are 90-something% macrophages, like 97% macrophages after this burn injury. Remember that there are no neutrophils that get across that airway in the burn-injured animals, but certainly none in the unmanipulated. And we could either take those cells right out of the animal and look for M1 and M2 markers by PCR, or we could culture them with the prototypic activators, either LPS as a stimulant or the combination of interferon gamma and TNF-alpha as M1 stimulants or IL-4 as an M2 stimulant. And one of the things that I should confess is that when you do bronchoalveolar lavage from a mouse, on a good day, you get 300,000 cells from an individual mouse. And on a bad day, you get 50,000 cells. So being able to do a lot of heroics of culturing in a variety of different stimulants is just not going to happen if you want to do studies on individual mice. And we want to do studies on individual mice without pooling these cells, so we're very limited in what we're able to do. But we are able to do some of these things. And if we look at IL-6 expression by those few alveolar macrophages that we're able to obtain, and we compare unstimulated cells, meaning medium alone, to LPS in vitro, we see that those alveolar macs produce whopping levels of IL-6 message if they are from young animals, and way less if they are macrophages from aged mice. And if we look at LPS stimulation of ARG1 as an M2 marker, even with LPS stimulation, we see some elevation. Again, not whopping levels, because here we're going up to 750 or so fold expression. We go up to 7 or 5-fold expression of ARG2, so it's really very diminutive. And if anything, we see an increase in ARG2, an M2 marker, in the macrophages from aged mice given LPS in vitro. And again, that could be a phenomenon of cell culture. We didn't have enough cells in these experiments to be able to do IL-4 stimulation. We have to go back and get more aged mice and do more of them to be able to do those comparisons, which are going on now. But in just looking at alveolar macrophages in the absence of any extra stimulation, just doing lavage on those animals and removing those cells, looking at 2 different M2 markers, ARG1 and YAM1, we see that the macrophages from the aged mice, just pulling out those macrophages and doing PCR on them, those produce way higher levels of M2 markers from aged mice than young mice. So we see higher levels of pro-inflammatory mediators as well, or I guess, do we see this? We've seen this in the mouse model. What do we see in elderly burn patients? And these studies are very preliminary, so I'm kind of worried about talking about them because I know you guys are going to make fun of me, because the surgical resident who's been working on this project didn't do everything exactly perfectly. Because those of you who've worked with surgical residents may or may not know that they kind of do what they want sometimes, and they don't ask mom for help. So in doing this, In order to be able to get approval to do this from our institutional review board, we had to use patients who were being intubated or had a tube down their throat for some other purpose. We can't just decide to do this on patients, I guess, for fun. So in doing this, we had to use our burn intensive care unit patients who either had inhalation injury, meaning smoke inhalation, or were suspect of having inhalation injury. And those patients underwent diagnostic bronchoscopy, which allowed us to look at what's in their lungs. And those patients were lavaged to remove material from their lungs to clear their airway to allow them to breathe better. And we were able to collect the bronchoalveolar lavage cells and fluid from these from these patients, and measure things in them. We also, in some cases, were able to obtain blood from these patients to be able to see what's going on in the blood, to see if it's really a good window of what's going on in the airway. And we'll see what the data look like. So in doing these studies, what the resident did is he decided to go, because he's a statistician nerd, he decided to make sure that there's an even number of patients in each age group. So he chose 25, 24, and 25 by putting the patients of these different age groups from 18 to 40 years old, 41 to 58, and 59 to 86. Not the best way to separate ages. And it depends on who you talk to. Somebody who is within their reproductive years would be, you could cut this in the middle and say everybody over 55 might be in the older group. Everybody between 55 and 70 is kind of old, and everybody over 65. So I'm not really sure how to shake this out, and we'll have to talk a little bit more about the best way to do this. But what he did was, this is what he did. He looked at our burn ICU patients first for their demographics, and then split patients just based, I guess, on this vague age criteria. And one of the things that he was able to look at is this concept called a Bow score, or revised Bow score. And this is an analysis of total body surface area burn, whether or not there's inhalation injury present, and comparing that to the patient's age. And by doing that, it showed that there were age-dependent differences. And most of the other parameters, as you can see from the statistical analysis, were not different. Looking at the outcomes of these patients, whether they passed away or how long they stayed in the hospital, were other clinical parameters that he examined. Sepsis was significantly different, but mostly because the middle age group didn't really have much sepsis, for whatever it's worth. Days that were free of the ventilator were very different, that the older population stayed on the ventilator for their entire time in the ICU, or whatever percentage of time he was showing here. And mortality was increased in the aged population, which again was not surprising to us. When looking at cytokines and pro-inflammatory mediators, we measured about 27 or 28 of these mediators in these patients. Here's data from plasma, and don't go nuts trying to look at everything. We found, thankfully, convincing evidence that the elderly population had higher IL-6 levels, and also had an increase in a twofold increase in IL-1 receptor antagonist, an anti-inflammatory mediator that sort of is one of the counteractors of IL-1. We didn't see differences in many other mediators, but randomly saw increases in eotaxin, which we can't really explain. That's used to just be an eosinophil recruiter, but now seems to recruit many other cytokines or many other cells. When looking at the BAL fluid for cytokines, among the things we saw was a marked elevation in MCP1, a macrophage recruiter and activator, which was elevated, as well as interleukin-8, which is a neutrophil recruiting mediator. So we see at least some things that we might expect, but also see an elevation, a significant elevation in IL-15. But if you look at the elevation in IL-15, it goes from 0 to 2 picograms per mil, not exactly something you'd want to write home about. But these other mediators, I mean, at least MCP1 going up from 35 to 240, and IL-8 from about 1,000 to over 2,000, might be reasonable elevations to want to write home about. What we also see, because these patients have an insult that comes from the airway, is that we see that there's a marked elevation in the percentage of neutrophils, different from our mouse model where we see no neutrophils in the airway because our mice are not subjected to inhalation injury. So we see up to about 80% of the cells obtained by BAL are neutrophils. So to summarize those observations relative to young burn patients with inhalation injury, older patients with comparable injuries spend more days on the ventilator and have a higher rate of mortality. There's about a twofold increase in plasma levels of a pro-inflammatory mediator IL-6 and an anti-inflammatory mediator IL-1 receptor antagonist. In the BAL fluid, we see about a fourfold increase in MCP1 in the elderly patients and also of IL-8, which I think was not quite that high. So we're still working on this M1, M2 phenotyping. Again, from BAL cells from burn patients, we get a lot more cells. We don't get just 100,000 or 200,000. We can get millions of them, but it's taking a long time to start collecting those, culturing those cells and trying to figure out their M1 and M2 phenotype, but that's one of the things we're working on. So in conclusion, like elderly burn patients, our old mice or aged mice are less able to survive a burn injury than younger animals. The aged environment with inflamm-aging might prime the neutrophils to have a more exaggerated response to burn injury, and this can be partially ameliorated by treatment with CXCR2 antibody. The splenic macrophage studies looking at aged mice show a global immunosuppression, while alveolar macrophages from aged mice seem to have an elevated level of M2 markers. So there's a tissue-specific or organ-specific difference. And we see having high percentage of M2 macrophages would render a patient at greater risk for pulmonary infection or pneumonia, which is an important problem in the elderly population, as Carlos has probably told you many times. So my parents modified their terminology to say inflamm-aging sucks, which may or may not be the case, but doesn't sound like it's a good thing to have. I'd like to thank the people in my lab who perform many of these studies. The aging studies are currently being done by Leah Brubaker, who's an MD Ph.D. student, and Jessica Remus, who's my lab manager. There are a number of other people from my former empire who worked on the macrophage studies, including another MD Ph.D. student, Eric Bamer, and yet another MD Ph.D. student, Vanessa Namalini, along with Christian Gomez and Shagufta Mahbub, who were both postdocs. The clinical studies were initiated by Jocelyn Albright, who's a surgical resident in the lab, continued by Chris Davis, and although I don't have an arrow there because he hasn't done a whole lot on this project, Stuart Carter, who's still in the lab working on these studies. We have a pulmonologist in our group, Aaron Lowry, who wrote a K23 grant application, which we hope to hear positive news on in the next couple of weeks. In addition, the clinical studies in particular, we had great help from our burn ICU staff, as well as my former chair and dean, who is now our provost, Rich Gamelli, who has been very helpful. Just about everything is done in collaboration with Mashkur Chowdhury, who is one of our long-standing collaborators over the years. In addition, Pam Whitty runs our immunology and aging program and has been very helpful, and my colleague Luisa Di Pietro, who helped with a lot of our cutaneous wound healing studies over the last God knows how many years. And of course the feds, who are my favorite F word, who have funded us pretty well to do many of these studies. So we've had funding from multiple NIH institutes, the Department of Defense, as well as a private foundation, which has now given us, unfortunately, our last chunk of money from them. We've gotten some funding also from the International Association of Firefighters. And lastly, I'd like to leave you with an advertisement, which is for the next Society for Leukocyte Biology meeting. While I am the past chair of the society, I'm now the head of the meetings committee, another dubious honor. And our next meeting is in Newport, Rhode Island in October. And one of the reasons that I bring this up is the abstract deadline is way far away. But also to let you know that there's going to be an awesome session, a plenary session, on leukocytes in the aging host, in which Carlos is a speaker and I'm also a speaker. So I encourage you to come to Rhode Island to attend that meeting. And thank you for your attention. I'd be glad to answer more questions if there are any.